Okay, good morning and welcome to the 2020 virtual edition of the Royal Tyrrell Museum Speaker Series. Today, the Royal Tyrrell Museum and its cooperating society are proud to present Dr. Karma Nanglu. Karma is the Peter Buck Deep Time Postdoctoral Fellow at the Smithsonian National Museum of Natural History in Washington, D.C. Karma is originally from Toronto, Ontario, and received his Bachelor's of Science degree from the University of Toronto. For his undergraduate thesis, he investigated coral communities in Wakatobi Marine National Park in Indonesia. Following his undergrad, Karma stayed at the University of Toronto to pursue a PhD in the Department of Ecology and Evolutionary Biology at the University of Toronto. His thesis investigated the diversity, ecological, and spatial temporal structure of the Burgess Shale paleo community. This research made use of the extensive Burgess Shale collections at the Royal Ontario Museum and also focused on fieldwork, collecting new specimens. This fieldwork included work at the Marble Canyon fossil locality, which is one of the recently found new really important Burgess Shale localities. In 2014, I had the opportunity of joining Karma for his fieldwork at Marble Canyon, one of the few times that I've been able to do Burgess Shale fieldwork. Following his PhD, Dr. Nanglu traveled to Washington, D.C. to take his current post at the Smithsonian. His current research focuses on an enigmatic area of the animal tree of life, the early evolution of a group of animals known as the hemichordates. Dr. Nanglu's fellowship also includes outreach and education, particularly related to the recently opened Smithsonian Deep Time Fossil Halls. And this is something he's happy to answer questions about as well. Today, Karma will be talking about some of his postdoctoral research on hemichordates, entitled Worms from the Rocks, Burgess Shale Fossils Shed Light on Old Mysteries. So without further delay, I'll hand the mic over to Dr. Karma Nanglu.
Great, thanks, Caleb. I can go ahead and share my screen now. If the thing will go to full screen. Let me just start by saying thanks to Caleb for that introduction, and thank you, Caleb and Francois and everyone else who was involved in inviting me to give this talk. Obviously this year and last year are particularly odd, so many of us aren't going to conferences or we're going to virtual conferences instead. And so being able to give guest seminars and lectures like this, I think really helps fill some of that void that I'm missing in terms of interacting with people and talking about the kind of research that's ongoing for me at the moment. And so as Caleb sort of alluded to, and as you can tell from this title, what I'm going to be talking to you about today are fossils from the Burgess Shale, in particular, some of these sort of vermiform, worm-shaped animals that are really quite mysterious, enigmatic in their evolutionary origins, and for which I think Burgess Shale fossils have recently been really pivotal in sort of uncovering. And so for a group like this, many of you, I'm assuming, are paleo enthusiasts having come to this seminar, don't need too much introduction to the Burgess Shale, so I'll go over this very briefly. This is a picture I took back, I think, in 2016 when I was up in the Burgess Shale. And so the Burgess Shale itself is a series of fossil localities that are all part of the Stephen Formation up in the Canadian part of the Rockies. And they were discovered almost about 100 years ago, a little over 100 years ago now, by Charles Doolittle Walcott, who you can see on the left. He was at one point the secretary of the Smithsonian. And so, having been able to work at the Royal Ontario Museum's collections of Burgess Shale material, which are ever growing and are the largest in the world, and now being able to work at the Smithsonian's collections of mostly Walcott's original collections is really, the perfect marriage of two data sets for me. So I can't say how lucky I've been. And so these fossils are about 506, 508 million years old. The estimates kind of vary a little bit. So just as a frame of reference, when thinking about most of the big conclusions we're coming to for the rest of this talk, we are stepping back about 500 million years in time for all of them. And so it's a lot of time to kind of wrap your head around, but it's something that you should sort of keep at the back of your mind when we're talking about evolutionary origins, major groups, and such. And so I threw this slide in here because I want to reinforce that work on the Burgess Shale is ongoing. And so a lot of people are introduced to the Burgess Shale through things like Stephen Jay Gould's book, Wonderful Life. A lot of people have read this book, including myself. It's fantastic, although a little bit out of date now. But we're still going back and collecting new fossils from the Burgess Shale. And so I am a huge proponent of going back to museum collections and looking at old collections, trying to dig out new information from there. And in fact, the first couple of studies I'm going to share today involve that type of work primarily. but we are still going up there. And so you can see these are some pictures of us actually clearing some overburden on the left. That's taking inventory of some of the new collections we've made from Marble Canyon, which Caleb just mentioned earlier. The picture on the left, actually the left and the center actually, are from the 2014 field season that he was talking about, that Caleb was at and Lorna O'Brien was at as well. And so they got sort of a front row seat for some of these new discoveries. In fact, it won't be the focal point of my talk today, but we still think that probably close to 25% of the species that we're finding at Marble Canyon may represent entirely new species. And that's after a further subsequent 8 years of work poring over that material. So quite exciting find, and these finds are still ongoing. On the on the far right of the screen there, what you can see is actually a helicopter with a long tether line, kind of getting ready to lift up a bunch of the materials that we're going to be bringing up to the Burgess with us. So you can hike up to a bunch of the sites, but in terms of bringing large-scale equipment that we need for a large field team for multiple weeks at a time, we need things like rock saws, we need jackhammers, we need other pieces of equipment. We also need like camping and tent materials, food and water for multiple weeks and large crews. Most of that we bring up by helicopter. And so I think, as you can tell from this, involving helicopters transporting large parcels up into the Rockies, it's a time-intensive process. And so what makes this all worthwhile? Why do we keep going back up there to these sites that have been discovered over 100 years ago in some cases? And the answer is the fossils, of course. They're absolutely beautiful fossils. So just at an aesthetic level, I think, hopefully, you guys have that same appreciation for these that I do. This is just a smattering of a couple of different animals that have been discovered from the Burgess Shale, and I want to hammer home two primary points here. The first is that what makes the Burgess Shale, both of them, I guess, are about what make the Burgess Shale so exceptional, but the first of that is size. And so if you look at this animal on the bottom right, Kootenai scolex, that's a polychaete from the Burgess Shale, which I described as part of my PhD. The whole length of that animal is only about 2 centimeters total. And so when you think about fossil deposits, you tend to think about large, very impressive looking things coming out of the rock, but the Burgess Shale preserves animals with such incredible fidelity that we get these tiny organisms preserved in exquisite detail. And that latter point, the fidelity of preservation, that exquisite detail of soft tissue anatomical characteristics, we can actually see in all of these fossils. So when you look at a typical fossil deposit, the kinds of things that tend to preserve, well, if you're in a marine setting from the early Paleozoic, you would think of things like shells, for example, the shells of mollusks, the shells of brachiopods, ossicles, so like the skeletal elements of echinoderms. And if you sort of go a little bit younger in time, people tend to think about things like dinosaur bones or the bones of other vertebrates, scales, teeth, things like that. Really the most recalcitrant parts of the body. If it's a relatively hard part of the body, and if it is a large part of the body, that tends to improve the likelihood of it being fossilized. In the Burgess Shale, not only are we getting these tiny animals preserved exquisitely well, but we're getting features like eyes, things like nervous tissue potentially, all sorts of soft anatomical features, tentacles and antennae, the kinds of things that you could never normally imagine being preserved. So if you follow my cursor here, for example, these are a pair of front-facing eyes. These are gill arches. These are, micrometer-sized little bristles that come off the body of this polychaete. If you've ever seen a polychaete before, you've seen all the bristles on their body, and we can actually tease apart individual bristles on a 2 centimeter long polychaete. That's the kind of preservation that we're dealing with in the Burgess Shale. And so that type of data, or that type of preservation, is not just valuable for the data it provides or the aesthetic beauty, but it tells us, or it allows us to make a lot of inferences about not just the evolutionary biology of these animals, but also their ecologies. And so these are some artistic reconstructions of those exact same animals that I was just showing you. And so, lots of these can be placed actually in the kind of environment or artist mock-up of the environment these animals lived in. And so when you have great preservation of morphological details, that allows you to then make inferences about the lifestyles of these animals, because we can't obviously see them actually in action as we would for a modern day animal. So we have to make those inferences based on morphology. The exceptional preservation of the Burgess Shale really helps with that. And of course, it allows us to sort of investigate the origins of these major groups by looking at how these characters will have evolved over time since their deep time roots. And so I like to hammer home the point that when we're studying the fossils of the Burgess Shale, the animals of this time period, we are in a very real way studying the deep time origins of pretty much every major animal group you can think of. The Burgess Shale, of course, is what I would still argue is the best window into what we call the Cambrian Explosion. That's a term I'm sure many of you are familiar with, but it refers to this rapid first appearance in the fossil record of pretty much every major animal group you can think of, pretty much every major body plan, as we call it, so major sort of suite of morphological characters. And so for things like arthropods, things like ctenophores, things like early chordates, sponges, we can find representatives of these groups up in the Burgess Shale. And so it's obviously been important, these fossil sites, for teasing apart the early evolution of pretty much every animal group you can think of. But I would say one major exception to that trend has been a group of animals called the hemichordates, which are the animals I'm going to be telling you about for the rest of this seminar. So who are the hemichordates? They're a group that I think a lot of people are not hyper familiar with. And that's understandable for a variety of reasons. One is that they're a little bit non-trivial to get to in modern day environments. And so I'm going to spend a little bit of time just introducing you to these animals. So there are two major groups of hemichordates. The first are called the pterobranchs. You can see some examples of them here. They're microscopic animals that have sort of tentacles that they use for feeding, which you can see kind of like here. Sometimes they're arranged almost like a basket or a crown. Sometimes they're arranged kind of like in a Y kind of configuration, like you see here. And they're microscopic, and not only that, but they live in colonies together. And so I think the animals are interesting in and of themselves, but I think when the story really gets interesting is when you consider that the lifestyle of these individual animals, which we call zooids, is facilitated by sort of attachment to each other, sometimes obligate, sometimes facultative, and the secreting of these homes that they build around themselves. So it's hard to tell from this frame here, but these homes are much larger than any individual zooid, and they live inside of them in a sort of shared way. You can think of it sort of analogous to coral, where coral is made up of, a coral reef, for example, is made up of lots of individual polyps that kind of share this overarching skeletal structure that they secrete together. In a sort of similar way, the pterobranchs will produce these collagen-based tubes, proteinaceous tubes, together and then live inside of them. And so, again, here's one of those zooids, the one with the sort of Y-oriented arms that I mentioned before. These are microscopic tentaculate animals, and they all live inside these colonies together. And so you can see each of these tubes has a little aperture at the top, and the zooids kind of poke their arms out and they reach out into the water column and feed on suspended organic material. I think, again, to draw an analogy here with a group that people are maybe a little bit more familiar with, it's a little bit similar to a crinoid, for example. In fact, this illustration, I think, really shows that nature quite well, in that you can see the arms, again, here are arranged as sort of a basket to capture water flow that kind of comes across of it. So a lot of paleontologists or paleo enthusiasts are quite familiar with crinoids, which have a fantastic fossil record. And so you can think of crinoids a little bit as your model when thinking about pterobranch feeding, or specifically this type of suspension feeding. And so these tubes themselves, I showed one example before, but they can come in a huge variety of different forms. And you can see all these little apertures out of which these animals would be poking out their arms. And actually, I would bet that a lot of you are far more familiar with pterobranchs in general, actually, than you realize right now. And that's because graptolites are actually pterobranchs. And so lots of paleos are quite familiar with graptolites, largely for their use in biostratigraphy, so the aging of rocks based on which animals you find in them. And so this is a sort of assemblage of different graptolites that I have on the left here. And so in some ways, pterobranchs have a fantastic fossil record in that we have graptolite tubes, spanning tens of millions of years. We have all sorts of different morphologies. They've been studied quite heavily, but they don't have a good soft tissue record. So their homes tend to preserve quite well, but we have no solid data about what the actual animals themselves would have looked like. Sometimes you have the hint of something that looks kind of like a little smear, or maybe some of the connective tissue in between where the individual animals would have been. But the animals themselves, we don't have any perfect fossils where, one sticking its arms out of its home, for example. And so I threw this in here because I think graptolites often get kind of a bad rap for being sort of boring. I hear it all the time. But when you imagine these as sometimes skyscrapers or apartment buildings filled with these animals that are building them and sharing the homes together, I think it becomes a little bit more exciting, at least. But as I said, without the soft tissue preservation of the zooids themselves, a lot of the graptolite biology is mysterious to us. And sort of these reconstructions that show the animals coming out of their homes are based on inferences that we make about the tubes. So those are the pterobranchs. The other major group are the entrepneus, which you can see on the left. People might be a bit more familiar with them. They're called acorn worms commonly. And this isn't the entirety of their fossil record, but up until relatively recently, this was the majority of it. They have an atrocious fossil record, both in terms of the age. So you can see that the oldest example I think I have here is from the Carboniferous. And that's not old enough to really get at the origins of the entire phylum Hemichordata. Most phyla, we get first good evidence of them coming into play around the Cambrian. So the Carboniferous is not as far back as we'd ideally like to go to look at the origins of this group. And most of these are not particularly well preserved. They're preserved well enough that we can recognize them as acorn worms or some form of acorn worm, sometimes the trace fossils of an acorn worm, but they don't have the kind of soft tissue details that you really want, a paleontologist really wants to make, rich inferences or robust inferences about things like the sequence in which different characters evolved or what characters were present in, ancestors to major modern groups, things like that, the pacing and timing of evolution. That's all difficult without soft tissue details and without internal details that are lacking in these forms. And so before I go on, I wanted to point out that at this stage, you might be thinking, how do we know that these two groups are closely related? They look very little like each other. And so I won't go into it in any great detail, but developmental data strongly supports this relationship. You can follow from the embryo all the way to the adult phase. You can see they have the same basic body architecture, the same sort of organization of their main body cavities, and also all of the molecular, like DNA-based data that we use to run evolutionary analyses also suggests the close relationship between the entrepneus and the pterobranchs. So we're quite confident in that, despite the fact that in their adult forms, they look very different from each other. But that disparity in how the adults look is actually the crux of a lot of problems. And so I just summarized here the characteristics of these two groups. So the entrepneus are worm-shaped, solitary. They tend to be infaunal, so they actually burrow into the mud, and they tend to be deposit feeders in modern environments. Whereas the pterobranchs are miniaturized, colonial, as I said, animals. They live sort of these shared tube dwellings together, and they're tentaculate suspension feeders. And so for evolutionary biologists and paleontologists in particular, often one of the questions we're interested in is, what did the last common ancestor to your group of interest look like? Knowing that, or at least having a good hypothesis about that, carries all sorts of implications for further analyses. You can start looking at related groups. You can look at things like the directionality of evolution within a certain group, gains and losses of characteristics. But that really is the underpinning behind a lot of that. And I think, or I hope you can sort of see the picture I'm painting here, that with the entrepneus and the pterobranchs being so massively different from each other, and with such a poor fossil record of each in kind of complementary ways, it's very, very tough to get at this question. It's been, I would say, mostly intractable. And so questions about what the last common ancestor may have looked like for Hemichordata have largely been theoretical rather than having much of a grounding in direct evidence from the fossil record. And so now that brings us back to the Burgess Shale. And so what I'm going to describe for the rest of this talk are a series of 4 studies that have taken place over about the last 7 to 8-ish years that I think are gradually helping to shed a bit of light on this quite enigmatic part of the animal tree of life. So the first study, and really the one that kicks off this series of studies, is a paper in 2013 by my PhD supervisor and his colleagues that took place actually before I joined the lab, and was the discovery of this animal Sparta branchis tenuis, which had been known for a long time from the Burgess Shale, but it was thought to be a different group of animals. They recognized it actually as a hemichordate, specifically one that shares a huge number of characteristics in common with the modern day acorn worms, one of which you can see on the right there. And so how do we recognize this right off the bat? The basic gross morphology of the animal is very easily recognizable. And so the brief primer here is that this number 1 indicates the proboscis. This is the anteriormost structure, the one right at the front of a hemichordate. It's kind of a muscular, mucus-laden structure that they use to feed and to burrow and do a couple of other things. We can recognize that very easily in these animals as well, in the fossils. Behind that, number 2 indicates the collar, which is a sheath of tissue that surrounds the mouth. And actually the mouth is located just at that juncture between 1 and 2 right here. And then posterior to that, number 3 is the trunk. So this is really what gives it its kind of worm-shaped aspect, which you can see very well along here. And it can be further subdivided, I'm going to give more details of this in a moment, into the pharynx, which you can kind of see the hints of right here. Those are actually gill pore openings out into the water. and then this posterior part of the trunk, which is mostly just an intestine. So we could recognize it right off the bat as being an entrepneuste or very closely related to them. This pushed back at the time the earliest record of the acorn worms by about 200 million years. So again, a big finding in and of itself. But to really highlight how incredible the Burgess Shale is, this photo on the left, I think that field of view is probably less than a total 1 centimeter. And you can see fine details like these striations, which represent the gill bars inside of the pharynx of this animal. And they look very similar to those that we find in modern day hemichordates. So that's that same species, Gensiglossus pusillus, but I've dissected it so you can see internally. And so in a lot of ways, if the study had stopped here, the conclusion you might have drawn is an incredible one, that there's been this huge amount of morphological stasis in this group of animals over the course of nearly 500 million years, which is pretty shocking. The most shocking part of the study, though, is where it starts to diverge from what we know about modern day acorn worms, in that this animal, Sparta branchis tenuis, actually built tubes around itself very much like a pterobranch. And so you can see the actual worm itself preserved inside here, but they build this tube around themselves, and I'll show a few more examples of these tubes. They can vary in their appearance. Sometimes they look very heavily corrugated. So B and C here are the same fossil, but under different lighting conditions. You can see this sort of poorly preserved worm inside, but then this large tube preserved exterior to it with this kind of corrugated structure, possibly for enhancing sort of rigidity of the tube itself. Presumably the tube has a kind of defensive structure or nature to it. But it can also take these very wild looking forms with branching structures like you see here. This really odd sort of spiral looking one. These are two-dimensionally compressed fossils, but we can still see many of these details. These odd ones that are ring shaped that seem to have no opening at all. I will just admit to this now, this is an area that requires a little bit more study, probably a follow-up study on just the variation in tube architecture in this animal. But we are finding, at the very base at least, acorn worm-like worms that live inside of tubes like their pterobranch relatives. And I'll go on, but before I get to that, I wanted to say that we also find never more than one worm in a tube. So that's an important distinction, because the pterobranchs live in their tubes in sort of colonies where there's more than one individual per tube, and they build this large tube around themselves together. As far as we can tell with Sparta branchis, it's one worm builds one tube. So to summarize the study and where we were at the end of it, which is just before I started my PhD, is that we have these two major modern groups that we have pretty good resolution for, and we have this one fossil here. So the conclusion that you might naturally draw is that the last common ancestor, represented by the star, may have looked something like Sparta branchis, in that it was vermiform, like an entrepneuste, but tube building like a pterobranch. That's a very neat story, and it feels intrinsically very satisfying. But of course, this is just one fossil, and this is a huge group of animals, not particularly specious, but as a major division of animal life. So one fossil obviously is not ideal. And this is kind of where my research starts to jump in. And so part of my PhD was redescribing this animal called Ousia disjuncta, which you can see on the bottom of the screen. And I'm massively summarizing this because Ousia disjuncta has had a very tortured kind of history of study. I'd be happy to talk more about that in the question period if people are interested in that. That's a talk in and of itself in terms of different researchers jumping in on this question. But what we found was that a re-examination of the Ousia material, as well as new collections from the Marble Canyon, which I talked about earlier, showed that actually it is a hemichordate. And again, we can see the same basic three-part body plan, very clear: proboscis, collar, and trunk. But there are two major differences in this instance compared to Sparta branchis, which looks almost like a modern-day hemichordate. The first, indicated by the number 3 here, is that the pharynx is extremely long compared to any modern hemichordate. And the second is this kind of claw-shaped structure that's at the posterior margin of the animal. So let's go to the pharynx first. So it extends about 85% of the way through the body. And these U-shaped gill bars, which I showed you before, extend that entire length. And so in most modern acorn worms, the pharynx, that region that has the gill bars, is only about 10 to 20% of its total body length. In Ousia, it's often about 85 to 90% of the total length. So right off the bat, extremely different from any modern hemichordate. And I think, honestly, that probably contributed to it not being recognized as a hemichordate for quite some time. The second part that's very weird about Ousia that I want to talk about requires me to take a quick divergence into something that looks totally unrelated, but it will all dovetail, I promise. So this fossil here, called Margaritia doris, which has since, I think, the 1930s at least, been considered to be some form of algae. You can see it's a long tube-shaped structure, which has these kind of pores that are ellipsoid, that are arranged in a spiral ring going up the length of the tube. It's quite large in that it can be branching. The largest actual fossil of Margarita that I've seen a photo of, I don't remember what location it was from, but it traced about 50 centimeters, so like half a meter in terms of its total length, which for the Cambrian is pretty gigantic. And it actually has quite a unique sort of construction to it as well, the morphology. So this fossil here, which again, you can see there's a bifurcation here. You can see these ellipsoid structures. But if you zoom in on one of these ellipses, what you can see in this specimen is that the construction of the tube itself is quite odd in that it's created by a series of like micrometer-sized fibers that interweave across the surface of the fossil, almost like a sweater, to produce the texture that you see. And the sweater sort of unweaves and then comes back together in order to form these ellipsoid pores. And so these arrows actually just point to a couple of nice examples of individual threads or fibers, quite complex in its construction. And the reason I've taken us on this kind of odd-seeming divergence is because with specimens from the Marble Canyon in particular, what we find are abundant tubes of Margaritia with actual hemichordate worms identifiable as Ousia preserved inside of them. This fossil in particular is, a good example of how lucky you have to be with things sometimes. It broke out of the rock perfectly. So the line I'm tracing here with my mouse cursor is actually a perfect break in the rock that reveals the worm, and on either side shows where the fossil, where the tube is. So this is the kind of thing where it would have been pretty difficult to prepare out with a micro engraving tool or something, but the perfect hammer split in the rock up in the Rockies worked out perfectly. So again, we can tell this is Margaritia. It has the fibrous elements. It's got these ellipsoid pores, not perfectly well preserved, but they're there. You can see the branching nature of this, although it's diverging down into the matrix of the rock here, and the worm preserved inside. So let me break this down into just schematic anatomy for a second, because there's a lot going on. Most of the time, you don't see the worm and the tube preserved perfectly. But if we kind of create a composite picture of what this would have looked like and break it down into its constituent parts, you can see that the tube itself here is highlighted in blue, and the worm itself is in black. And so again, I want to point out these weird extended pharynx that runs the entire length of the body, and this posterior structure at the back, which presumably has some ecological function related to its lifestyle in the tube. So to sum up, we have these large tubes with the worms inside, and we think that this tube-shaped dwelling suggests a relatively sessile lifestyle, not unlike some of those modern-day pterodactyls I showed you earlier. If you're spending a lot of energy producing this gigantic structure that can be up to half a meter long, it makes sense to spend most of your life in there. It's a huge energetic expense. This unique extended pharynx, we think, suggests a sort of suspension feeding niche. And I want to point out that not suspension feeding in the same way as, for example, the modern day pterodactyls, which capture water with their arms. So not quite like that, but more like some of the modern day entrepreneurs that suction water into their mouths and pass it through their bodies by generating current. And the way they generate current is with little hairs called cilia that are on the U-shaped gill bars themselves. And so these kind of beat in time and draw water in. And so we think that this really long extended pharynx would have just increased the capacity for creating that unidirectional water flow into the mouth. If you are up on your inverts, this is maybe a little bit more analogous to how like a tunicate, for example, would feed with its large pharyngeal basket, drawing water in and then pushing it out of a secondary siphon. So in this context, then the pores inside of the tube make a lot more sense in that it can facilitate water and gas exchange into and out of the tube. When you have tube building organisms or even animals in modern environments that build burrows, you tend to want some area for interchange between water inside the tube and water outside. Otherwise, it can get quite stifling inside of there. And so the pores would have facilitated that. And we think that the tubes themselves were perpendicular to the substrate, which will allow for more exploiting of the water column. And to that point, this posterior bilobe structure, which is shaped almost like a claw, we think would allow for some vertical movement up and down the tube. The tubes, as I said, can be up to half a meter in length, but the longest worm that we found is only 10 centimeters. And so if you're going to actually fully exploit all of that extra water column that you're getting by building the tube upwards, you need a way to move up and down that tube, which is not easy when you have a worm-shaped body plan. So we think that that appendage may have helped with that. So we now have a second study, which in some ways reinforces the conclusions we had from the first. We do get more support for tube building being ancestral to this group. We do get more support for a worm-shaped body plan being ancestral and not the zooid body plan of the pterobranchs. But what we're also seeing is that in the Cambrian for hemichordates, like almost every group that you can study, there are aspects of their morphology that don't have a nice, clear, neat analog in modern biodiversity. So this really underscores the fact that without fossils, with just a survey of modern day biodiversity, getting at questions of ancestry or what kind of characteristics would have been present early on in the evolution of a group are really quite speculative. So the third study, and the last one that I'll go into in depth, regards this animal, Gensiglossus senis, which we just recently described. It came out last summer. I named it actually after my dad. His name is Genze. And so going over the anatomy of this animal, just from anterior to posterior, we first have the elongate muscular proboscis, again, very much like this modern day Saccoglossus. These long proboscises tend to be used for feeding on sort of seafloor, deposit feeding, where they kind of scan it over the seafloor and they use the cilia and mucus on these proboscises to sort of siphon mud back into the mouth. But behind that, we have this crown of feeding arms that's very much like a modern day pterobranch. And so starts off looking like an acorn worm, then it looks like a pterobranch. We move back one step, and again, we have this vermiform trunk, again, like an entrepneust. And so you can see the kind of hybrid nature of coming into clear sort of resolution here. But before I get to that, there's this odd structure at the end, which almost looks like an anchor or a ballast, which doesn't necessarily have a neat clear analog in the modern day hemichordates. But we do find this kind of posterior appendage in both Ousia, which you see here, where it's, again, divided into kind of like a bifurcating claw. And then in Sparta branchis, where it looks very simple, again, like a ballast structure, as we see in Gensiglossus. So when we look at the modern biodiversity to try to come up with an analog for this, or even better, a homolog, something that has the same sort of shared ancestry, one thing you can find is that by looking at the developmental biology of some of the acorn worms, they actually do have a posterior postanal tail as they're developing, which they reabsorb as they grow into adults. And so positionally, it's very similar to what we see in the fossils. And this tail is used kind of to adhere to the benthos sort of submarine muds that they might be on. You can imagine these animals are quite small, so currents might be able to easily knock them away. So there might be a bit of an attachment structure or function behind that structure. That structure has in turn been suggested to be homologous or share an ancestry with this structure here called the stalk. It kind of looks like an elephant trunk, which we find on the pterobranchs. And these stalks allow the actual members of these group of a colony to attach to each other. So it's a feature that facilitates their colonial lifestyles, and it's positionally, at least, somewhat similar to where we find these postanal structures in the fossils. So this reconstruction was done by an Ontario-based paleo artist named Emily Damstra, and she did a fantastic job, I think. And it's actually a great time to talk about ecology. So first of all, we have this crown of feeding arms, which obviously makes sense to be used in suspension feeding as it does in their modern day relatives. And so we've suggested that suspension feeding was the primary mode of life for this animal. But it also has this very elongate muscular proboscis, which again, in modern day entrepneus is used for deposit feeding. So we've suggested that this animal might have actually had a sort of bimodal ecology in that it could both suspension and deposit feed at different points. And then in terms of this postanal attachment structure, again, we've suggested it might be for both attachment and motility, particularly attachment to the sort of seafloor, and now allowing this animal to kind of sit more upright, a little bit more analogous to what we see right here in the pterobranchs, the kind of posture that you'd want if you are trying to exploit the water column directly. So with these three described, we were able to run some phylogenetic analyses. So these are analyses looking to produce a tree like this that tells us which animals are related to each other most closely and in what order. So I won't get too far into the weeds there, except to say that we do find that the acorn worms and the pterobranchs form two neat groups, which you find here in purple and in blue. Sparta, branchis, and Ousia are what we call stem group Entrepneus. So they're not within the biodiversity of modern day Entrepneus, acorn worms, but they come out more closely related to that group than to any other. So they're quite illustrative about understanding the early evolution of the acorn worm specifically. Gensiglossus comes out as a stem group hemichordate. So in this position, stem group to the entire hemichordata. So by contrast, this is a little bit more illustrative, or we might look to this fossil for broader insights about the group overall. And so what is the emerging picture then that we have? We do have some data suggesting then that tube building is plesiomorphic or ancestral for the crown group hemichordates, so all of this group here. What I've highlighted blue are all of the animals within this group that actually build these secreted tubes around themselves. So you can see it incorporates most of the early branches. Also, post-anal attachment structures appear to be ubiquitous among these early branches. And so what I've highlighted now are all of the sort of earliest representations or representatives rather of the hemichordates. And these three studies in total, merging the paleo data with what we know from modern day biodiversity, suggest that the last common ancestor of this group was vermiform, that tube building was ancestral, at least for all of the animals up to this point. And that, as I said, these post-anal attachment structures, which are not particularly well represented in the modern day acorn worms, were actually probably ancestral for the total group, and they seem to be present in all the Cambrian forms that we find. So before I draw this all together, I wanted to point out, now we're at our third study. We have another fossil, which again, if we'd found this first, we would have drawn a completely different set of conclusions. And so it does support a kind of worm-shaped body being ancestral to the group, but you can see that it's a hybrid of the Entrepneus and pterobranchs in almost the opposite way as our preceding two studies, in that it has the worm-shaped body plan of the Entrepneus, the tentacles of the pterobranchs, but it doesn't share the tube-building lifestyle with the pterobranchs. And I'll further complicate this issue by throwing one sort of series of figures in here about a research study that is actually ongoing. And so this is another tube from the Burgess Shale, relatively large. It's probably about 20 centimeters, I think, in total length, this one. I've highlighted it here because it's fairly faint. But if we zoom in on just one area and highlight it, you can see that there are a bunch of polychaetes in here for one thing, but also there are vermiform hemichordates preserved inside of these tubes. And so we know that vermiform hemichordates build tubes in the Cambrian, despite the fact that they don't look like they should based on our understanding of their modern relatives. And what's really important about this study is that we find, I think we found close to 30 or 40 of these tubes, and they often have more than one hemichordate preserved inside. And so you'll remember with Sparta branchis, with Ousia, they were worm-shaped, they built the tubes around themselves, but there was only ever one worm per tube. Now we have tubes with multiple worms inside, possibly living together, probably in my mind, constructing the tube together in a way that's analogous to the colonial lifestyle of the modern day pterobranchs and presumably the graptolites. So again, we have a new simple vermiform hemichordate in a giant tube with colonial living possibly. And rather than show you just a series, again, another one of those trees where it looks very neat, like the progress of different characteristics being acquired, what I want to the way I want to frame this is that hemichordates have been sort of critically understudied in this time period, largely because we haven't had the material for it. And as we now start to unravel what's possible for this group at that time period around the Cambrian, near the origins of the phylum, we're finding that every combination of possible characteristics you can imagine was present. Early in my PhD, when I was thinking about these questions, I remember writing down in one of my notebooks just a bunch of possibilities for what the last common ancestor may have looked like based on what we know of their modern relatives. And I had a bunch of scenarios involving different character combinations that I thought were possible or likely, less likely. And as I've gotten to work on more and more hemichordates, I've come to realize that essentially we're finding representatives of all those combinations. So again, we have their modern acorn worms, modern pterobranchs, major morphological differences between them. And as we start to flesh this out with fossils, we're finding combinations of major morphological and ecological traits that fit almost exactly as we would have hypothesized them. But as we add more and more fossils, it's not unilaterally clearing things up. It's giving us a better idea of what it was present during the time. But what looks like what was present during the time was every sort of combination conceivable. Perhaps that shouldn't be surprising when you understand or when you spend some time looking at how odd the animals of the Cambrian are. But this is all to say that while a bunch of questions about the last common ancestor are clearing up, we still have many outstanding questions. And in like any research program, as you study more questions are developed. And so I don't want to end on a note like we just don't know what's going on, because we actually have gained a much greater appreciation for some questions that have been lacking in direct evidence historically. And so this question, to return to, what did the last common ancestor look like? This question that, is a driving, motivating factor for many paleontologists and lots of evolutionary biologists in general. For the hemichordates, the emerging picture seems to be of a vermiform animal, not like a pterodrank, that there was a postanal attachment structure present in the last common ancestor of the group and possibly present in all representatives of the group in the Cambrian. And it also seems as though tube building may have been ancestral to this group, although the lack of a tube in Gensiglossus does muddy that water a little bit. So what I hope I've convinced you of over the course of this talk is that hemichordates are interesting on their own merits, and I really think they are. And I think every animal group is interesting on their own merits once you get to know them, although it took me a little bit of time to be convinced on this group of animals. But if you need a bit of a broader context to realize why this is important, all you have to do is take a couple steps back in terms of the breadth of the relationships we're talking about from an evolutionary standpoint. So that question of what did the last common ancestor look like to the hemichordates, that question is relevant to what the last common ancestor looked like between the hemichordates and their closest relatives, the echinoderms. So that question right here, which is in turn related, these are nested problems, to the last common ancestor of our group we call the deuterostomes, which includes the hemichordates, the echinoderms, as well as the chordates. And of course, we're included among the chordates. I said earlier that when we study the fossils of the Burgess Shale, we are studying the deep time origins of modern day biodiversity in a very real way. And I think when you kind of take a broader look at things and you look at these vermiform fossils for a long time and you start to understand their evolutionary relationships, I think what you realize is by studying these worms, our distant cousins, we're actually getting, a bit of an actual look at our shared ancestry with a huge diversity of different animals that you wouldn't think are related to chordates and to humans at all. So that's a bit of the broader pitch there. And so with that, I'm happy to take any questions about the research that we've talked about or the Burgess Shale in general.
Great. Well, thank you very much for that, Karma. We already have a couple of questions on our little Q&A thing. If you're at home and you have questions, feel free to type those in the Q&A and we'll get to those with some time. The first question is about Ousia, and they would like you to please elaborate on that complicated history you mentioned at the beginning.
Actually, as I said that, I realized what I should have done is I should have included that the secondary series of slides. But essentially, Ousia, so it was part of some of Walcott's original publications back in the early 1900s. I think in 1911 is when he described a bunch of the sort of worm-shaped animals from the time period. They were all lumped together in a publication, and one of them was Ousia, and he suggested it might be an animal called a polychaete. And so essentially those structures called the gill bars that run through the body give the body a sort of very vague feeling like it's been divided, like it's a series of striations that are divisions within the body. And that is a characteristic of the annelids, the segmented worms, things that include modern day earthworms, the bristle worms like Kootenai which I mentioned earlier. And so Walcott's original suggestion was that it might be a kind of animal called a polychaete. So that's where he left that. In the 1920s, I think 1920 itself, a German researcher named Lohmann suggested that it might actually be a tunicate. So in the end, much closer to where we ended up ending with this animal than was the current conversation at the time, but that didn't gain a lot of traction. It's very different looking from a tunicate for one thing. And then in the 1960s, Conway Morris, who many of you would be familiar with if you studied or if you've read Wonderful Life, suggested that it's not a tunicate, it's not an annelid, we just don't know what it is, so it got lumped into that group, the Enigmatica, one of these Cambrian forms that we just can't parse out yet. 2005, a researcher suggested it was a type of worm called an arrowworm, to which Conway Morris replied it was not, to which the other author came back and suggested, yes, it is, and that's where everything got left off. That's the kind of field that I was in when I jumped in on that project. And so the first chunk of the study was essentially just disproving all those previous hypotheses. And if I had a tree to show, across the tree of life, the amount of different spots this animal has jumped to, considering it is one of the more innocuous fossils from the Burgess Shale, it could not be more different. It's moved between, I think, 3 major divisions of animal life, including one of the most confusing, those arrowworms, over the course of the last 100 years. I am assuming that we got it right now, and that I'm quite confident that we have it right. Although, that might be the focal point of a conversation 70 years from now, and I'm just the next in a sequence of people to have gotten it wrong. Which, science is iterative, so I wouldn't mind.
Sounds good. The next question is, given the gap between the Carboniferous fossil and the Burgess Shale examples, can we rule out convergent evolution?
So convergent evolution on that sort of same vermiform body plan is how I'm interpreting
Yeah,
that.
presumably that these are convergent in that body plan.
Yeah, I think, convergence is like the... is the underlying problem when studying evolutionary biology in general, is like the perpetual problem. In fact, there wouldn't be so many evolutionary biologists if convergence wasn't such a problem, because then there'd be far less to study. So yeah, I think there's some possibility there for sure. When you have a major gap like that, it's hard to make any inferences. So where I feel like the Burgess Shale fossils really have a lot to say is looking at the spread of characteristics that were sort of possible near the dawn of this major group of life. But I didn't want to go much further than that and start integrating fossils from later, knowing that there was such a massive jump in quality for such a large period of time. So who knows how many flips back and forth between a vermiform and terrabranch lifestyle may have been possible. That, I think, becomes doubly true when you look at things like Gensiglossus, which have tentacles, but also a vermiform body plan. There may have been all sorts of sort of intermediary forms or sort of other lineages that we just don't have recovered. We don't exactly know what the graptolites look like. So presumably, based on the way their tubes are built, they looked like modern-day rhabdoplurids. Those are the zooids that look like their arms are Y-shaped. But now that we have these tubes where we have long worms preserved together, possibly building the tube together, I'm not sure that that's even completely true either. So I wouldn't say it's something that I would rule out, especially given the massive gap in time between the Burgess Shale and the next really solid examples of soft tissue preservation.
Okay. Our next question is, the soft body fossils that you described are exquisitely preserved, and they're presumably bottom-dwelling organisms. The degree of preservation would reflect an anoxic depositional setting. So how do these macroscopic organisms, which would require a fair amount of oxygen, end up in that anoxic environment?
Yeah, so the conditions, you're right that they would either be, the conditions around the fossilization itself would presumably be anoxic or at least dysoxic, moving towards anoxia. So part of that scenario is the actual submarine mudslides themselves. So I didn't have time to talk about the actual depositional setting and such of the Burgess Shale, but these animals were deposited in their positions by submarine mudslides, which were relatively rapid, which would have buried them, possibly transporting them some distance. And that would have helped create the anoxic conditions as sort of like oxygen directly around the animal itself, around the animal being preserved, was kind of snuffed out. And then they are kind of sealed under there, if that makes sense. Sealed might not be the exact right word. I think it brings in other implications, but it's kind of like entombed, I guess is the better word. And that helps create dysoxic situations. There's also the possibility of being pushed towards a more dysoxic environment, but presumably, as you said, these animals needed oxygen to live in their primary environment, not necessarily the one in which they were fossilized.
This one gives you a chance to speculate a bit. What new fossil would you most hope to find in the context of your research question? And have you speculated about what it might look like?
That's a good question. I'm kind of surprised I never come up with a ready-made answer for that, because I'm always asked, and I feel like every time I give a different answer. And I think that reflects the fact that I would just die for anything that could help me piece out the order in which these things occurred. I think the question I was asked earlier really strikes at something important, in that obviously I work on the Burgess Shale, and I love the Cambrian. I think my headspace will be there for the rest of my career, but it would be nice to get a couple more fossils of this quality from younger time periods to see whether or not some of these morphologies had some staying power to them, or they really were things that sort of blinked into and out of existence. I would love to find fossils of terrabranchs that actually have soft tissue preservation. So some graptolite tube that actually has the animals inside of them would be a dream for, I think, many people that worked on graptolites as well. Some direct evidence that they actually had the arms that we kind of presume that they have based on the way the tubes are built would be fantastic. Another thing that's possible is I'm really getting focused on that area. So this is like really in the hemichordate weeds now, but there's the proboscis, which in the graptolites is transformed into kind of like a stumpy structure that builds the tube. It secretes it kind of like laying down concrete or painting a fence almost. It kind of produces collagen fibers. Right behind that is the feature called the collar, which in acorn worms is just a pretty simple sheath and sort of it siphons mud into the mouth pretty much. But that unit of the body is where the arms are developed in the pterobranchs and presumably the graptolites. So I would love more fossils with greater details of that area that show the different kind of morphologies that might have been possible. And I've been recently thinking about this because some of the deep sea acorn worms, if you guys aren't following like Noah and Mbari on Twitter, you should, because those videos are incredible. And, it's great background stuff for when you're working too. They've shown that some of the deep sea acorn worms have these long kind of like arms extending off of this area called the collar. So in a group that typically doesn't have any elaborations there, they have these kind of like, they're arranged almost like a chevron. And so the purpose behind them might be to expand the surface for deposit feeding, almost thinking like a snowplow, kind of pushing through deep sea muds or catching sort of like deep sea, like, organic snowdrift. So I would love any fossil that shows more details of that. Honestly, I'll take anything at this stage. I have a strong feeling that in a lot of old collections, there are hemichordates that have just been put into unidentified worm, or like Ousia, its tube was put as some kind of algae because it hadn't gotten maybe some of the attention that it required, and we hadn't found all the fossils needed to piece that together. That was a long answer because I'd love to find all sorts of fossils.
The next question is about the illustration or the reconstruction of the Ousia tubes. Mm-hmm. It looks like the end of the tubes were closed. Is that what's that based on?
Yeah, no, that was well spotted. So the ends of the tubes are the worst preserved parts of the thing because most of the time they're broken. So again, if you can imagine these submarine mudslides, presumably these kind of like tall skyscraper-like structures, a lot of them will have snapped. We often don't get perfect preservation of them. And often we can tell they're damaged because as they become damaged, if you imagine just like, tearing your sweater or something, the threads in that area start to unravel. It's not like the entire sweater unravels at the same pace. That break allows a sort of, like a margin along which the thing is more progressively damaged. We see a very similar thing in the fossils. So most of them we can tell are broken because they're kind of unwoven at both ends. We found a few that look like they come to a closed end. So we kind of use that as the basis of our reconstruction, that the top will be closed. But again, I would say that's not like a definite thing at this moment. We are making that inference. But it's, it's possible we'll find ones where like there's a nice kind of almost like a chimney, kind of like a sponge where it's open. And we have seen that in Sparta branchis where it looks like the worm is actually crawling out of the tube, which has led to that different interpretation.
Okay. We have another question. This one is about the process of naming a new species. Can you just describe what's involved with that?
Yeah, so the first thing obviously is you have to find a combination of, so generally speaking, there's two components to the name. There's the name for the genus, which refers to a large group of closely related organisms. And then there's the species name itself. So there's those two components. Typically, you want to choose things that are somewhat informative about either the group of animals it belongs to or some aspect of its morphology or possibly where the animals were originally discovered or something about the range of locations you can find them in. So using Gensiglossus, for example, that was an example where I wanted to name something after my father, who was the inspiration for me to get into science when I was a kid. And so that's the first part, Gense. Glossus, I believe, is from either Greek or Latin, I forget which, for tongue, which is a common suffix for this group of worms. They used to be called tongue worms occasionally, although I think another group has superseded them and are now known as tongue worms. And then senes is from Greek senex for old, or Latin senex for old. So it's an old worm belonging to the group of worms that typically have the suffix glossus.
The next question is, can you speculate as to whether the animals you're talking about could have been like modern day barnacles in the sense that they attached themselves to either rocks or other animals, other organisms, I guess?
Yeah, I think that's very, very possible. And so We don't have any, so we actually do have evidence of all sorts of animals from the Burgess Shale that do look like they're directly attached to other organisms. So for example, brachiopods attached to the spines of some mollusk relatives like Ousia. That's pretty definite. We have brachiopods attached to sponges, for example, but you're going to notice a trend here. These are animals that have like an obligate attachment rather than a facultative one, which we're suggesting for these types of animals, where they could kind of use it almost more like ballast than a true full attachment. So I think while it's possible, the animal itself is relatively decently sized for the time period. So I don't imagine it, crawling onto another animal and then, standing on its shoulders or anything. So I think it's more likely that it was just attaching to the benthos. So you're correct about that. For the direct attachment, they tend to be more animals that are truly sessile, that don't move around at all. Things like most brachiopods, some mollusks, things like that.
Okay. Why are they called acorn worms? There's apparently no resemblance that this person sees.
It really comes down to the proboscis. So that first and that anterior most structure, some of them, they're kind of short and squat. They are just shaped like an acorn. There's really no more to it than that. But the ones I tend to feature aren't the ones that look like that, because I think it's just easier to visualize when you have a long proboscis. Also, it's more analogous to what we find in that one particular odd species, Gensiglossus. And it's also just the best photo I have of a hemichordate, but it's because of the proboscis shape.
The next question is, have you done any work comparing other Cambrian sites or lower Ordovician sites, such as the Feijoada Shale?
Yeah, so I have not yet for other time periods. We have compared across multiple sites. And so actually the thrust of my PhD thesis was not about these worms specifically, but it was about paleoecology. And I think Caleb alluded to that in his introduction. And so one of the big parts was describing the paleoecology of the entirety of the Marble Canyon, how these animals would have lived together, how they changed over the course of thousands of years rather than millions, which you can normally get from looking at like a locality to locality kind of scenario. And then taking that data and integrating it with what we know from other sites, including Walcott's original quarry, which was the focus of my PhD supervisor's thesis, the tulip beds, which was the focus of actually Lorna O'Brien's thesis, and the Raymond quarry, which is a series of fossil sites that are just above the Walcott quarry. So we have looked across sites, and I think we don't find Gensiglossus in multiple sites. We find Sparta branchis primarily in one, and a little bit in the others, and Ousia the same, but kind of reciprocally. So there's no one Burgess Shale site so far where it looks like all of the hemichordates are there in great abundance. It seems like Walcott mostly has Sparta branchis, Marble Canyon mostly has Ousia, Raymond quarry has very few of either, and tulip beds has very few of either. That animal Gensiglossus comes from a totally different location. Actually, Raymond quarry has a bunch of that new species that we're describing with the large tube and multiple worms inside.
Okay, there's only a few questions left here. One is about new technologies, particularly for imaging, and if there's any developments that might, I guess, allow you to get more information from the fossils.
Yeah, for sure. So like scanning electron microscopes are not exactly new technology, but it is a piece of technology that was not available to Walcott, for instance. And a lot of the fossils I focus on, if I was to be asked in a professional setting, like what kind of research do you focus on, I would say, origin of phyla and body plans, paleoecology, but really a lot of that comes down to, I like to tackle the groups that people ignore. So a lot of the fossils I work on haven't been studied in decades or close to 100 years, like Ousia. And so SEM for some of these things actually is new technology when applied to those fossils. We've also done a little bit of elemental mapping. So looking at the sort of topography of a fossil in terms of what elements are preserved in what locations, and whether or not we can tie that back to any patterns we see in the anatomy of the animals themselves. So I don't want to oversell that as though, we find aluminum in this area, and every time we find a high density of preserved aluminum, that means it's like the brain of the animal or something like that. But if we find a similar preservation profile across fossils, that can tell us that maybe those the parts of the body that have a similar preservational profile in terms of the elements there may have had a similar original tissue type that decomposed or broke down into constituent elements in a similar way. So we have used that for Ousia. They didn't make it into the publication, but we've started to look at that a little bit. But none of those results are really out there yet. We're not exactly sure what to make of them.
And one final question, and that is related to fieldwork. How do you decide which material to bring home to collect for the museum? What's the process involved with that?
Yeah, it's a tough one. I think part of it is when you have a large group out there, you have lots of people with different expertise, and they can sort of individually weigh in for their group. Like, if one of the worms comes out, I can run a quick eye over it. And, in the field, they don't often look as good as they are going to once they've been cleaned up and prepared and such. But someone like me can give a quick pass and say, that looks like a good worm fossil because I've looked at hundreds and thousands of them and I have a sense for it. Same thing for arthropods, same things for other groups. So part of the determination is that. Hopefully, like a bunch of these fossils are going to make it into the Royal Ontario Museum's Dawn of Life gallery, which I think will hopefully be opening up at the end of 2021. And so some of it is done with aesthetic or display reasons in mind. So there's a science reason, there's the aesthetic reason. Those are the two primary ones, shoring up what we have lacks of in the collections. So often, we won't necessarily need to collect en masse animals that are very well represented in the collections already. So we'll just take back the greatest examples of those. But also something that I think factors into our decision-making is when we go up to the Burgess Shale, at least the way the ROM crew tends to do things. I'm sure lots of other crews do a similar thing, but we take note of everything that we don't collect and have to be tossed. And so when we make those ecological analyses, like I was mentioning for Marble Canyon, that's based on as complete of a censusing as we can possibly have for that site, not just what makes it back to the museum. And so we would have a massively biased analysis if we only looked at what had been collected, because often that is for, the unique, perfect example of a new species, or for, something that's going to eventually go into an exhibit. That being said, I think in terms of total numbers of pieces of rock, which range from things that are this big to pieces of rock that are the size of your table, we brought back probably on the order of maybe 2,000 or something, representing in total, when you count all the individual animals, something on the order of 22,000 specimens. So believe it or not, we are choosy, but we bring back a lot. It's hard to say, it's hard to not bring them back. They've been waiting for us for 500 million years, and we're all very attached to them.
Sounds good. Well, thank you very much for that really, really exciting talk. And thank you at home for your attendance and for your questions. Don't forget to join us next week when Dr. Ryan McKellar from the Royal Saskatchewan Museum will be talking about his research using amber to investigate dinosaurs and their habitats. Okay. Thank you very much.
Thank you.